So here's another video on amino acids in the amino acid video series, and this one is on special amino acids. So there are four special amino acids that come in the category of special amino acids due to their unique properties. So proline, glycine, histidine, and cysteine, these are the four amino acids that we'll talk about, and these are usually discussed in the category of special amino acids due to their very weird or unique properties. Okay. So we'll start with proline and glycine. These are called alpha helix breakers. So they are rarely ever found in, usually not found in alpha helices. Alpha helices is the secondary structure found in most proteins, and we'll talk about alpha helices and beta strands in the protein uh, video series. Okay, and why is proline and glycine? Why are these two alpha helix breakers? Let's talk about that. So proline, it has a really weird R group which is fused to its N terminal, right? So every other amino acid we know has a um, N terminal which is separate from its R group, right? When we usually draw the amino acid structure, how it looks is that it is actually like that, like this. So we have an N terminal, right? And here's your R group, and we have a C terminal, and here's your hydrogen atom, right? So we know that N terminal, whenever we draw amino acid structure in its generic form, the N terminal is separate from the R group, right, as we know it. But for proline, it's actually your R group is fused with the N terminal, so it causes kinks in the alpha helices, right? It causes steric hindrance, so it promotes bends due to this steric hindrance causing R group, which is fused to the N terminal. It causes kinks, it causes bends in alpha helix or secondary protein structures. So what is usually found, where proline is usually found, is that it's found in bends, what we call beta bends. So these are beta bends, and these connect. This beta band is a short string or chain of amino acids that connect to anti-parallel or parallel beta strands. So we'll talk about beta strands and alpha helices later in protein video series. Okay, but for now, just remember that proline is actually found in bends. It's not found in alpha helices. Glycine is the same thing. Glycine's R group is also weird. It's just a small single hydrogen atom. And due to that, it minimizes steric hindrance, right? Because there's no bulky R groups as in proline or in tryptophan. Those amino acids shouldn't be found in alpha helices because they cause kinks. Tryptophan also has a bulky R group, right? Two rings of two carbon rings in tryptophan, which causes a lot of steric hindrance. So it's not found in alpha helices. Glycine is also not found in alpha helix because it is too flexible, right? We want some steric hindrance to cause a helix to form, right? But glycine is too flexible. It is also found in beta bands. So it's also a beta band favoring amino acid. So proline and glycine, they're alpha helix breakers, but what we normally call them in a positive manner, we call them band formers or beta band formers or beta band favoring amino acids, right? And also another thing to note about glycine is that it has two hydrogen atoms, so it doesn't have a chiral carbon, right? So glycine has no isomers. All other amino acids have D and L isomers. Okay, so that's what's up with proline and glycine. Histidine is also really interesting amino acid. Let me talk about that. Why? Because it's amphoteric. It can act as an acid as a base as well. Why? Because histidine has two nitrogen atoms in its R groups, right? And which have their lone pairs. So they are really good bases, right? We know that amines and amides, those are really good organic bases. So histidine has the same thing. It has two nitrogen atoms instead of just one, and both have their lone pairs. So they can act. It can act as a really good base, but it can also act as an acid. Why? Because it's the pK of the R group is just 6.04, right? Which is really close to the physiological pH of 7.3. Water is a really good example of an amphoteric substance. We all know that it can act as an acid and a base as well. And since histidine pK is so close to a pH of normal pH of or physiological pH of 7 or 7.4, it can also act as an acid or a base. So any pH below this 6.04 value of its R group pKa, it will accept, it will be protonated. So in that case, it will be able to donate protons, right? So it will act as an acid. Histidine in that case will act as an acid if the pH is below its pKa, R group pK of 6.04. So according to that, this Henderson-Hasselbalch equation, which I talked about very much in the PI calculations and net charge calculation videos, there, there the main theme was everything depends on the Henderson-Hasselbalch equation. This is really important, right? But if the pH is greater than this pKa, if there is a basic environment, then what will happen is that these nitrogen, this R group or these nitrogen atoms will be deprotonated, like I've shown over here in this form, right? So it's in its normal form, how we write histidine normally, right? So if pH is higher than pKa, we know that according to this equation, the it will be in the deprotonated form. So in that case, it will act as a base. So histidine acts as an acid and a base. So it's amphoteric. And let's talk about cysteine at last. So cysteine is so important that it's actually found in almost all of the proteins found in our body. Why? Because it forms the really important disulfide bridges, the SS bonds, right? These two sulfur atoms or two cysteine amino acids come together in oxidizing environment, and they lose their hydrogen atom because oxidizing agents, what they do is that they remove hydrogen atoms, right? They remove electrons. So what happens is that two cysteine amino acids will come and they'll shake hands and they'll form disulfide bridges. Two sulfur atoms will form uh, a bond in between them in oxidizing environment. Okay. So why these disulfide bridges are so important? Because they stabilize the tertiary structures of proteins, right? So that's the idea. And to stabilize tertiary structure, we need these disulfide bridges. So that's why cysteine is so important. It's found in almost all of the proteins, right? Because without tertiary structures, protein cannot function properly. They at least need to be in their tertiary form. And I'll talk about all these alpha helices and beta strands, tertiary structures in protein video series. But that's the entire idea about special amino acids. There are more of them. Methionine is really important as a start codon. All the protein amino acid sequences start with methionine because methionine actually comes from the AUG. And we know that from translation, from a translation knowledge, AUG is the start codon, right? And AUG codes for methionine. So all of your proteins actually start with methionine. So methionine is actually also a really important amino acid. But these four are in kind of a spotlight when we talk about special amino acids. So I'll talk about translation in those videos as well in molecular biology series. Okay, but that's the whole idea about special amino acids.